Hello my dear friends, welcome back to CSIR at home. In this video, we can discuss the most important topics for CSIR examination. The first one, that is enzymes. In enzymes, 4 types of questions. Statements, match the following, plots and problems that we have already discussed in our previous video. Take the book and go to the chapter Enzyme Kinetics, then read it. Okay, read only the heading and subheadings. Then you will get all the statements related to enzymes. For example, the formation of enzyme substrate complex is the first step in enzyme catalysis. Substrate concentration can affect the rate of enzyme catalyzed reactions. So, study these subheadings. Then, match the following. For that, you have to study the enzyme classifications. Okay, that is oxidoreductase, transferase, etc. And the type of reaction and examples. Then, problems. For that, what you need to study? KCAT, KM, all those things you need to study. Okay. Then, plots. For that, you have to study competitive and non-competitive inhibition, etc. Okay. And analyze all the plots. Then cell cycle regulation. It's very important. You need to study CDC, CDC2, CDC28, then CAK, V1, etc. All the proteins and enzymes associated with cell cycle regulation. Then cell cycle checkpoints. Okay. And also link cell cycle regulation with replication and cancer. Replication. Replication, what you need to study? You need to study the basics of replication first. You can study it from carb. Then the enzymes involved in replication. Okay, DNA polymerase, helicase, etc. Then study the structure of DNA polymerase. That is very important. Then you can study replication origin from Watson, Bruce Alberts and Cooper. Then the mechanism in replication fork and DNA synthesis at replication fork. Okay. Then what you need to study? You need to study about topoisomerates. You can study it from Watson. Then fidelity of replication. These are the main topics in replication. The fourth important topic is cell signaling. We have videos on cell signaling. Just watch it. Okay. GPCR, insulin, RTK, MAPK, kinase pathway, etc. Okay. Developmental biology. That is very important. Signaling pathways associated with developmental biology. Okay. You need to study RTK signal transduction pathway. Then JAK-STAT pathway. Hedgehog family. Then VND family. Okay. And signal transduction pathways. It's very important. You can refer Gilbert 12th edition. In plant physiology, the most important topic is photosynthesis. You need to study the light reaction, C3, C2, C4 and CAMP cycles. Blood clotting cascade. In animal physiology, you can study first week, December first week, just start studying blood clotting factors. Before that, just study, get an idea about basics of blood clotting. Then study blood clotting factors. Then study blood clotting cascade and disease associated with blood clotting. In genetics, you can study problems. Okay, do as much as problems. You can study epistasis, pleiotropy, sex-linked genes, dosage compensation, etc. from Gardner and Benjamin. Then study shell coiling of snail. Animal diversity, that is also an important topic. You have to study larval forms in invertebrate. First, we can study Tornaria larva. It is invertebrate. Okay, you can refer Kotpar. Tornaria larva. Then you can study Echinodermata larval forms. Orcularia, Doliaria, Bipinaria, Echinoploteus larva, etc. Okay. Study the larval forms of invertebrata. Here you can study Echinodermata and Hemichordata. Okay. 2 class. Echinodermata and Hemichordata. In ecology, study Lotka-Volterra competition models. you can study the different type of competition, then Lotka-Volterra competition models, and 4 consequence of Lotka-Volterra competition equations, then competition among Paramecian species. You can refer Peter Stilling. Then you can study mutualism, commensalism, predation, etc. In evolution, study species concept and pre-zygotic isolation. Study from Mark Ridley. This is very important. Agrobacterium mediated gene transfer. This is one of the important topic, the most important topic for CSIR examination. So you have to study the basics about Agrobacterium mediated gene transfer, then TI plasmids, then tDNA transfer, then Vir genes. That is very important. Okay, Vir genes. Then co-integrate vector, binary vector, that is pea green, for example. Study all the things, okay, in Agrobacterium mediated gene transfer. You can refer Primrose and BDC. From methods, you have to study immunological methods, RIA, ELISA, etc. Okay, you can refer Rob Reed and also study NMR. That's also very important. If you have followed our previous timetable, just revise all those things, okay? Or if you are new, just study, no problem. If you are just starting only, no problem, just start studying, okay? These are the topics, these are the important topics. We can discuss the timetable for day 1. Morning 5 to 6 a.m. study unit of replication, basics. Then 6 to 7 a.m. cell cycle regulation. We have videos on it. Okay, 2 videos. Just watch it. Then 9 to 10 a.m. study blood clotting, which book to refer Tortora. Then 10 to 11 a.m. light harvesting complexes, segrantes. Then 11 to 12 p.m. we have enzyme kinetics. We have so many videos. Just watch it. Okay. Playlist biochemistry. Check on the playlist biochemistry. 11 to 1 p.m. just watch the videos on enzyme kinetics. Then 3 to 4 p.m. competition. Study from Peter Stilling. Then 4 to 5 p.m. cell signaling basics. Watch the video. And you can also refer Leininger. Okay. Leininger, acetylcholine receptor and cell signaling. Okay. Basics of cell signaling. You can refer Leininger. Then 6 to 7 p.m. developmental biology basics. Just study the basics from Gilbert. Then 7 to 8 p.m. genetics. Reference book Gardner and Benjamin. You can study epistasis. Then 9 to 10 p.m. study Agrobacterium mediated gene transfer. You can watch the video. We have video on Agrobacterium mediated gene transfer. Or refer Primrose page number 277. Then 10 to 11 p.m. study the immunological methods from Rob Reed. Then day 2, that is December 2, you have to study, the morning you can study the replication enzymes involved. Book Watson. Okay. Then 6 to 7 a.m. study cell cycle regulation from Carl. 9 to 10 a.m. blood clotting factors, Tortora. Then 10 to 11 a.m. organization of light absorbing antenna system. Then 11 to 12 p.m. study previous year. Okay, just work on the part A previous year questions. Okay. Then 12 to 1 p.m. enzymes, Lippincott illustrated reviews. You can read the chapter. Okay, it's very easy. Lippincott, it is very easy. Or you can refer Stryer. Okay, just read the subheadings and headings. And also just analyze it. Okay. Then 3 to 4 p.m. Lotka-Volterra competition models. Then 4 to 5 p.m. insulin, cell signaling insulin. You can watch the video and make notes. Or you can refer Leininger. Then 6 to 7 p.m. developmental biology. Developmental biology is very important topic, signaling in developmental biology. So you can study RTK signal transduction pathway from Gilbert 12th edition. Then 7 to 8 p.m. pleiotropy Gardner. Then 9 to 10 p.m. tDNA transfer Primrose. And 10 to 11 p.m. methods. Okay. Then December 3rd, you can study the structure of DNA polymerase enzyme, CARB. It's very important. Okay. Then 6 to 7 a.m. cell cycle regulation, Bruce Alberts. Then 9 to 10 a.m. the blood clotting cascade. Then 10 to 11 a.m. mechanism of electron transport, Z scheme. Then 11 to 12 p.m. mechanism of electron transport. Then 12 to 1 p.m. enzymology inhibition, graph, study the graph. Lineature, page number 209 and 210. Then 3 to 4 p.m. Lotka-Volterra, Peter Stille. Then 4 to 5 p.m. GPCR, cell signaling. We have 2 videos on GPCR, study it. Or you can refer lineature. 6 to 7 p.m. Developmental biology, signaling, JAK-STAT pathway. Then 7 to 8 p.m. sex-linked genes in humans. Then 9 to 10 p.m. Vir genes. This is very important, okay, Vir genes. Then 10 to 11 p.m. NMR. Then December 4, we can study 5 to 6 a.m. study helicases, okay. Then 6 to 7 a.m. cell cycle regulation, mechanism of action of 2 DNA damage checkpoints, CARB. Then 9 to 10 a.m. sickle cell disease, hemophilia. 10 to 12 p.m. C3 cycle. Then 12 to 1 p.m. enzymology previous year questions video, okay. Just watch it. Then 3 to 4 p.m. competition among Paramecian species, Peter Stille. Then 4 to 5 p.m. cell signaling diagrams, that is 4 form of intercellular signaling, Bruce Alberts. Then 6 to 7 p.m. hedgehog family, okay, developmental biology. Then 7 to 8 p.m. dosage compensation of X-linked genes, Gardner. Then 9 to 10 p.m. co-integrate and binary vector, and you can also study the examples of vectors. Then 10 to 11 p.m. larval forms, Torneria larva, okay, Balanoglossus, Codpal, Invertebrate, page number 862. Then day 5, December 5, replication origin, you can refer 3 books, okay, Watson, Bruce Alberts and Cooper, okay. Just make notes from it. Then 6 to 7 a.m. cell cycle regulation and replication, Watson, Lodish. Then 9 to 10 a.m. revised blood clotting. Then 10 to 11 C2 cycle. Then 11 to 12 p.m. just work on the previous year questions on part A. Then 12 to 1 p.m. biological species concept, okay, and ecological species concept, Mark Ridley. Then 3 to 4 p.m. mutualism and commensalism. Then 4 to 5 p.m. CAM kinase. It's from Bruce Alberts, cell signaling, okay, CAM kinase 2. Then 6 to 7 p.m. development biology, VND family. This is also important, okay, VND signal family. Then 7 to 8 p.m. genetic maternal effect, shell coiling of snail. Then 9 to 10 p.m. leaf disc transformation, Primrose study from Primrose. Then 10 to 11 p.m. echinodermata larval forms, Auricularia and Oleolaria. Then December 6, you can study replication for Watson. Then 6 to 7 a.m. just do this previous year questions, okay. Then for 9 to 10 a.m. topoisomerase, Watson. Then C4 cycle, 10 to 12 p.m. C4 cycle. Then 12 to 1 p.m. phenetic species concept. Then 3 to 4 p.m. predation. 4 to 5 p.m. RTK, Bruce Alberts, cell signaling, okay. Then 6 to 7 p.m. VND signal transduction pathways, developmental biology, Gilbert. Then 7 to 8 p.m. genetics question practice, just do as much as questions. Then 9 to 10 p.m. Agrobacterium mediated gene transfer, you can study it from PDC. The same thing, okay. Then 10 to 11 p.m. echinodermata larval forms, Bipinnaria and Brachiolaria. Then day 7, December 7, morning 5 to 6 a.m. DNA synthesis at replication for Watson. Then 6 to 7 a.m. revised cell cycle regulation. Then 9 to 10 a.m. fidelity of replication from Karp and Cooper. Then 10 to 11 a.m. CAM cycle. Then 11 to 12 p.m. part A previous year questions. Then 12 to 1 p.m. pre-zygotic isolation, Mark Ridley. This is also very important, okay. Then 3 to 4 p.m. revised ecology, 4 to 5 p.m. revised cell signaling, and 6 to 7 p.m. developmental biology, revise it. Then 7 to 8 p.m. revised genetics, 9 to 10 p.m. revised agrobacterium. 10 to 11 p.m. you can study echinodermata larval forms, Ophioploteus larva and Echinoploteus larva. This is the timetable. So if this timetable is very hard for you or you can't study this one, just study some from it. Okay, this is 13 important topics. You can study 6 to 7 important topics from it. Okay, you can choose. So there is 2 months for your examination. So start study. If you like this timetable and topics covered in this video, please like and share so that others also got benefited. Thank you.